Hi, I'm Dr. Divyashree Gangaraju. Today I'm here to explain the concept of probiotics and how it got evolved. The term probiotics comes from a Latin word wherein the pro in the probiotics means for, or it can also be called as before or forward, and bios in probiotics stands for life. Therefore, any compound or molecule which comes with for life property or life promoting property can be viewed as probiotics. But in the context of microbiology and applied biotechnology, we use the term probiotics in order to refer to a beneficial microorganisms which when ingested in adequate amounts confers various health benefits. I'm sure all of us know more of antibiotics than probiotics. So, as the name says, antibiotics are the compounds or the drugs which comes against the life property, unlike probiotics, which comes with for life property or better life promoting property. When we take antibiotics, what they do is they have a detrimental effect on your gut ecology. They kill most of your gut microflora. However, probiotics, being the beneficial microorganisms when ingested, they help to prevent your gut ecology and gut diversity, and they are the ones which brings peace to your gut. Generally, when we say bacteria or when we think of bacteria, we what it's our mind is that these as a disease causing agents. But do you guys aware our body is made up of full of bacteria which comprises of both good bacteria and bad bacteria. Generally, we say good bacteria as the beneficial microorganisms and the bad bacteria as the detrimental microorganisms. In order to have a good health or the good better gut health, there should always be a balance between good bacteria and the bad bacteria, what we call it as eubiotic gut or eubiosis state. This achievement of eubiosis state is very much important. Why? Because this state helps to train and regulate our immune system. But when there is an imbalance between good bacteria and the bad bacteria, or when you have more of bad bacteria in your gut, it leads to either overactivation of the immune system or underactivation of the immune cells. Now, to give you an example of different types of good bacteria and the bad bacteria which reside in human GIT, you can refer to this slide. The examples of the good bacteria which resides in the intestinal tract or in the colon include the genera of bifidobacteria. The bifidobacterial genera are the most prominent ones which are found in human GIT. These are the bacteria which play a major role in digesting the ingested prebiotic fibers. Once they digest these prebiotic fiber, they aids in the release of important metabolites which contributes to specific health effects in the host. In addition to its role in digesting the ingested fibers, they also play a major role in absorption and the production of important vitamins and minerals, and they also contribute to good gut ecology. We also have other bacterial genera which is most predominantly seen, which is lactobacillus. The lactobacillus belongs to a category of a bacteria called lactic acid bacteria. They're called lactic acid bacteria because they produce lactic acid as a major metabolic end product. Once this metabolite is being produced in the gut, it lowers the intestinal pH, because of which what happens, it reduces the growth of the detrimental or the pathogenic microflora. In addition to bifidobacteria and the lactobacillus, we also have Escherichia coli as a normal microflora in the GIT. The examples of the bad bacteria includes Campylobacter, which is widely associated with food poisoning. We also have genera of Enterococcus in the human GIT. Not all strains of Enterococcus are reported to be pathogens. We have Enterococcus faecium, which is available as a probiotic microorganism in the market. However, few strains of Enterococcus, specifically Enterococcus faecalis, is reported to be an opportunistic pathogen, and it is widely associated with hospital-associated diarrhea and hence classified as a detrimental or a pathogenic microorganism. The another example of the bad bacteria is Clostridium difficile, which causes severe diarrhea, especially after antibiotic use. This slide will give you an overview of GIT and its role in hosting bacteria. It starts with mouth and esophagus. Mouth can be considered as a primary site for digestion, and from the mouth the food gets pushed to stomach through esophagus, and esophagus can be viewed as a muscular tube which aids in the transport of the food between mouth and the stomach. The pH of the stomach generally varies between 1.5 to 3, and this is the site where the digestion of the macromolecules occur. Once food reaches the small intestine, wherein the pH varies between 4 to 5, the absorption of the monosaccharides, fatty acids, amino acids takes place, and the intestine comprises of duodenum, jejunum, and ileum. Each of these play a major role in digesting a particular ingested food. Then the food reaches large intestine, which has colon, ascending colon, descending colon, transverse colon, and even sigmoid colon, each playing major role in the digestion of a molecule or a food. And the pH of the small intestine varies between 6.8 to 7.2, and this is the site where absorption of the bile acids, vitamin B12 takes place. Please note, this is the site where most of your probiotic microorganisms reside, and this is the place where gut microflora is, which comprises of both good bacteria as well as bad bacteria. As you see, or as you come down the lane of intestinal tract, you see increase in density of the bacterial load, and this bacteria will account up to 1/3 of the mass of the fecus, highlighting its abundance in the gut or a colon. Now coming back to the definition of probiotics, the probiotics are the live bacteria, specifically lactic acid bacteria. Examples include lactobacillus, lactococcus, streptococcus, which produces lactic acid as a major metabolic end product. So now you know the advantages of producing lactic acid as a metabolic end product, right? Upon being produced in the intestinal lumen, it reduces the intestinal pH, thereby making the conditions not favorable for the growth of pathogenic microorganisms. That is how they maintain the gut ecology, by reducing the growth of the pathogenic microorganisms or the detrimental microorganisms. The other bacterial group is bifidobacteria, such as Bifidobacterium bifidum, Bifidobacterium longum are the few examples of this genera which are probiotic in nature. We also have selected yeast strains such as Saccharomyces boulardii, which are classified as probiotics. Therefore, these probiotics are live bacteria and yeast which improve your intestinal microbial balance through one of the different mechanisms, thereby offering beneficial effects on the host. Today, we have a universally adopted definition given by FAO and WHO jointly. FAO is Food and Agriculture Organization, along with WHO, which is World Health Organization. Jointly, they have given the universally adopted definition for probiotics, which is defined as live microorganisms, which includes both bacteria and yeast, which when ingested or administered in adequate amounts, they confer various health benefits on host. Please make a note here. This phrase adequate amount is very much important because you won't be able to see the health benefits if you are not ingesting the adequate amount. Now let us see how the concept of probiotics got evolved. It all started during the era of Antony van Leeuwenhoek, who showed us the existence of invisible world. Antony van Leeuwenhoek is considered as the father of microbiology. He was the first scientist to observe the single-celled organism using a simple microscope, and he named these single-celled organisms as animalcules. The scientific community later found that these single-celled organisms are nothing but the bacteria. In the year 1676, it could be the budding yeast as well as the human red blood cells. It was nearly 2 centuries after Antony van Leeuwenhoek's observation of the single-celled organisms on the microscope. It was Louis Pasteur, the French chemist, who found out that these microorganisms comes with a biological activity. What do I mean by biological activity here is that the microorganisms, when they grow as a part of their metabolic activity, they produce metabolites which might be toxic in nature. So these toxic metabolites might contribute to the development as well as progression of a particular disease, which is clearly postulated in his germ theory of diseases. And he is the scientist who is known as the father of modern microbiology, who has made the remarkable breakthroughs in the causes and the prevention of the diseases, and developed the first ever vaccines against rabies, anthrax, and also invented a process of sterilization, what we call it as pasteurization, in order to achieve hygiene and sterility. The concept of probiotic got emerged during Elie Metchnikoff, who is considered as the father of probiotic concept. In one of his book, which is titled as The Prolongation of Life Optimistic Studies, which is published in the year 1910, he hypothesized theories on human longevity, wherein in this book he postulated that the human aging can be viewed as a biological phenomena, and this is correlated to the presence of toxic substances present in the bloodstream. Now, from where are these toxic metabolites coming and then reaching the bloodstream? It is because whenever you have a gut dysbiosis, meaning whenever you have an imbalance between the good bacteria and the bad bacteria will overgrow and then results in the production of the toxic metabolites, which later enters the bloodstream. And because of the toxic substances present in the bloodstream, you see enhancement in the aging process. And he is the first scientist who considered aging as a biological phenomena, and he correlated this aging phenomena to a biological intrinsic factors such as gut microbiota. Based on this theory, he observed that the Bulgarians who consumed the milk fermented using lactic acid bacteria, remember a few minutes back we discussed about lactic acid bacteria, right? They call lactic acid bacteria because they produce lactic acid as a major metabolic end product. And this lactic acid has ability to maintain the intestinal pH and also have the ability to downregulate the growth of the pathogenic bacteria. Now, based on this theory, Elie Metchnikoff, who said that the aging is a biological phenomena and is linked to an intrinsic factors like gut microbiota, he observed that the Bulgarians who consumed the milk fermented using lactic acid bacteria showed better life or long life. And he attributed this effect these due to the presence of the lactic acid in the fermented milk. Now, based on this observation, he himself started consuming this fermented milk, which is fermented using Bulgarian bacillus, which was earlier known as Bulgarian bacillus. Today, it is known as Lactobacillus delbrueckii, subspecies bulgaricus, which is used as a starter culture in order to ferment milk into yogurt. Now, based on his observation, the physicians began to prescribe the fermented milk, fermented using this Bulgarian bacillus, to their patients in order to have a clinical outcome. Later, it was Hull and his colleagues who isolated the specific strain of lactic acid bacteria, what is called as Lactobacillus acidophilus. The functionality of this strain is that it has ability to completely metabolize the milk sugar. What is the milk sugar? We all know the milk sugar is lactose, right? And most of us suffer from lactose intolerance. Therefore, when we develop any fermented product using this lactose degrading bacteria, that is Lactobacillus acidophilus, this will solve us or give us a solution for lactose intolerant people. This was identified and discovered by Hull and his colleagues, and in the year 1899 it was the scientist Henry Tisser from the Pasteur Institute. He was successful in isolating a bifidobacteria from a breastfed infant, and he found that this bifidobacteria was more prominent in the breastfed babies, and he also observed the clinical benefits of giving this strain to a patients who were suffering from severe diarrhea. In the year 1917, it was scientist Alfred Nissel, in the outbreak of shigellosis. He isolated a specific probiotic strain called Escherichia coli, isolated from the fecus of an healthy soldier, and named this strain as Escherichia coli Nissel 1917. Even till today, it is available in the market to treat acute GI infections, particularly salmonellosis and shigellosis. In the year 1923, the scientist Henry Bullard made an important observation on the individuals who consumed a special type of fermented tea. He observed that the individuals who consumed the special type of fermented tea showed a less incidence of diarrhea, and this was observed during the cholera outbreak. Further, he went on to isolate the organism which was used for fermenting tea, and he was successful in isolating the fermenting microorganism, which he later found out that the organism is a special type of yeast, what is called as Saccharomyces boulardii. Till today, the organism is available as a probiotic strain and it is used to treat various digestive disorders. The first ever description of the probiotics was given by the two scientists Lilly and Stillwell in the year 1965, and they used the term probiotics in order to describe as the substances produced by the one microorganisms and which has ability to stimulate the growth of the other microorganisms. Please make a note here, Lilly and Stillwell gave the first ever description of probiotics as the substance and not as the microorganism. Later, it was modified by another scientist called Parker in the year 1974, who included the word organism for this description, which was originally given by Lilly and Stillwell. Parker gave the description or redefined the probiotic description as the organism and the substances which has ability to contribute to the intestinal microflora. The actual definition of probiotics was first brought out by the scientist Roy Fuller in the year 1989, who gave the definition of the probiotic as live microbial feed supplement which beneficially affects the host animal by improving its intestinal balance. So remember, this new definition removed the word substances which was originally included by Lilly and Stillwell. Moreover, this Fuller's definition emphasized the requirement of viability for probiotics and introduced the aspects such as beneficial effect on the host. Lately, by the scientist Jules Tormont, he expanded the use of probiotic concept not just for humans but also for other animals. And this probiotic concept for animal nutrition gained more popularity during the year 1980s and 1990s in the Europe, which led to the implementation of European regulation for probiotics use in the animal feed and gained more official recognition of probiotics that led the way for other countries. However, today, as I said earlier, we have a universally adopted definition for probiotics jointly given by FAO and WHO in the year 2002, which states that probiotics has the live microorganisms which when administered in adequate amounts confers various health benefits to humans and other animals. Just to summarize the concept of probiotics and how it got emerged, we started with Anthony van Leeuwenhoek, who showed us the existence of invisible words, what we call it as single-celled organisms in the compound microscope. And it was through Louis Pasteur, through his cell theory or the germ theory of diseases, we found out that these microorganisms comes with a biological activity, and if this microorganism is detrimental in nature may contribute to the development or the onset of a particular disease. The concept of probiotics was originated during the era of Elie Metchnikoff, who is considered as the father of probiotic concept. Henry Tisser in the year 1899 made an attempt to isolate bifidobacteria from the breastfed infants, and he was successful in isolating this organism, and he also showed the beneficial clinical outcome against diarrhea. In the year 1917, it was Alfred Nissel who isolated E. coli Nissel 1917 from a fecus of healthy soldier. The probiotic yeast such as that is Saccharomyces boulardii was isolated in the year 1923 by Henry Bullard. If you say the definition or the description for probiotics, which was originally given by Lilly and Stillwell in the year 1965, included just the word substance produced by one microorganism which has ability to stimulate the growth of the other, which was modified in the year 1974 by the Parker, who included the term organism in addition to the substance to a definition. And in the year 1989, Roy Fuller gave the first ever actual definition of the probiotics as the live microbial feed supplement, which has ability to improve the intestinal microorganism. And in the year 1919 to 1998, Jules Tormont expanded the concept of probiotics to animal nutrition. So with this introductory class on probiotics, I would like to conclude my session by saying the famous phrase by Hippocrates, who said that the all diseases begins in the gut. Thank you for exploring the origins of wellness. See you in the next class.